Okay,
Thank you.
sure. So, copy. Oh no, why won't let me paste this? Okay. Give me 1 second. I don't know what the hell's going on here. Oh, okay. Okay, so that's the information that does. All right. So all those things will be on the lab exam.
Thank you.
Sure. Hold on. I'm gonna put that in your audioverum as well, obviously. So okay. Does that make sense? So is there anything on that list that you guys don't understand?
So for the shapes and the stains of the specimens, the E. coli, Staphylococcus, Streptococcus, are you going to give us another like piece of paper with the slide or a tissue slide, and then we have to determine which one it is?
No, it's not that complicated. Yeah, it's not that complicated. It's honestly pretty easy. So let's see what is it? Come on, hurry up.
Professor Poon, is this going to be on Connect?
Yes.
Okay.
So the lab exam is on Connect?
No, the lab exam will be in person next week. Okay.
Okay, thank you.
The lab will basically be finishing up this lab. So you guys will be analyzing the results from your urine analysis, and then the lab exam, and that's it. Okay. Does that make sense?
Yes.
Okay. So from what I can tell, the average for cohort 1 was something like 87, 80. It's above an 85. So it's probably either low 90s or high 80s. Okay, so I expect you guys to do the same. So and that's really all I told them to. So it's not really too difficult. Okay. All right, does that make sense to you guys? Yes. Okay, cool.
So for our lecture exam, is it just the Connect practice questions?
Yes.
Okay, and do you recommend like focusing on the practice exam or the specific weeks of practice questions?
Here. Hold on. So, I would say both, but let me show you what your exam is. Your screen. So this is your exam. Okay, so randomly draw questions. So 29 out of 50, 27 out of 59 questions from chapter 11, 29 out of 57 questions from chapter 12, 13, and, you know, so everybody will have more or less different questions. There's very little overlap. And then there you go.
Cool, thank you.
Sure.
Professor, can you scroll up just a little bit?
That's it. Like this. That's it.
11, 12, 13, and 14.
Yeah.
I thought we were also doing 15, so only through 14?
Yeah.
Okay.
Who the hell is Remy Red? Oh, okay. Okay. And any other questions? Like anything in these chapters are difficult? No? Okay. No, that's it. We're done. That was all you needed.
Do you recommend we should focus on anything?
I honestly, I don't know because your choice of questions are random, right? So it's not like everybody gets the same test. Does that make sense? So like, and everyone gets very different tests because like you're choosing something like half of all the questions. So there's gonna be a lot of individual questions for each one.
Professor, can we go through some identifying some microbes?
Like how?
I'm not quite sure, but it's just one of the lists that you showed us earlier. So there's going to be like one of those multiple choice.
Oh, okay. On Connect, right? So let me see. Week 9, week 8.
By the way, what was the bacteria that we worked with today?
There was like 3 or 4 things in there. I mixed 3 or 4 things in there on purpose.
Oh, okay.
So when you stain it, what you saw was the pink was very much E. coli. I put a lot of E. coli in there. So there's E. coli in there. I'm pretty sure the purple was Staph epidermidis or Staph aureus. Both of them were actually in there as well. And then I forgot I put one other one in there, but I don't remember what it was.
Okay.
Does that make sense?
Yes.
Good.
Thank you.
So yeah, if you guys did the gram staining properly, which some of you guys did, you guys saw a mixture of purple and purple and pink colonies, right? Okay. especially Jay. I think I remember Jay did it right. So yeah. Yeah. All right, cool. So okay. So which one do you want to see? So phenotypical methods of identification. Oh, identifying white growth. Okay, got it. Okay, that was the most question. Okay, so you can basically it's telling you can identify microbes different ways. One is genotype the whole thing. You just look at it like so now that sequencing is incredibly cheap. It takes no effort at all to pop your bacteria into the genome sequencer, and the results come out in an hour or so roughly, and you can sequence the microbe that way. I think that's the most modern one. You can also look at the phenotypes. So and most labs still do this. They do gram staining, and then they follow basically the dichotomous key. And if it's gram positive, then you do X. If it's gram negative, you do Y. And eventually you come up to with the result of what the bacteria is. That is actually the bunch of labs that you will do week 9 and week 10. It's all this. It's the same lab over and over again. You find an unknown bacteria and you have to figure out what it is. Okay, and you guys have a whole bunch of labs to do that, and everyone is different. So, yeah, and then the last one is immunologic, which means you use antibodies that you know work, and you know they work to which bacteria, and therefore, if the antibody binds, then that's the one you have. Does that make sense? Okay.
Thank you.
Yep.
Did you say
Professor,
how many questions are on the lab exam, Dr. Poon?
50.
Okay. Thank you.
are you gonna make a post in this week's discussion board so we can respond to the peer reviews?
I will do that right now. Thank you for reminding me.
Thank you.
Yes, I still have. Six. Discussion board. All right. All right. What else do you guys need help with? Discussion.
Any chance we can go through some questions, just like how we did a review from last term?
Sure.
Thank you.
All right. Oops.
If you could do one with the blood, just to refresh my memory, like the maternal fetal blood thing, blood typing.
Okay. Okay, let's go over that really quickly because that seems to confuse a lot of you guys. So okay, so what are the 4 possible blood types?
A, B, AB, O.
Okay. All right. So A, B, AB. So this is the donor. This is the recipient.
I'm not seeing anything, Professor. I'm just seeing a white screen right now.
Yeah. Oh, Okay, no worry.
Oh, sorry. Thank
I'm just writing a bunch of stuff. Which okay. All right. Now you guys see this chart, right?
Yes.
Okay. So yes or no?
Yes.
Yes.
No.
Yes.
No.
you. No. Yes.
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 No. Yes.
Really?
Yes. Doesn't it?
I'm just going off what you guys are telling me.
Well, I thought that those were no's. Because it's A, B, 2 giving to A, right? And B, no.
Well, AB, I'm sure you guys all really can give it to AB. Right? And then
No.
no. Okay. And this whole column is
Yes.
right. All the way through. Okay. Does that make sense to you guys? Okay. So the other component of blood is whether or not you're Rh positive or Rh negative. Okay. So can a Rh positive donate to a Rh negative?
Yes.
No. So plus, minus, plus, minus. So obviously this is a yes. Right? And obviously that's a yes. Right? So can the donor give it to the receiver? What goes in this box? And what goes in that box?
Wouldn't that just be positive still?
They're not mating. So if I have Rh negative blood and you have Rh positive blood, can I give you my blood?
No.
No, they will all have to be RH negative, right?
Mm-mm.
Yes.
If I am Rh negative, then I'm not going to be able to receive Rh positive blood because that would look like a foreign antigen to me. Does that make sense? If I am, okay, if I am Rh positive, then if I get RH negative blood, I won't even see it. My immune system won't even recognize it at all. There's nothing to recognize. Does that make sense? The plus is the only thing you can recognize. Does that make sense to you guys?
So Dr. Poon, what would happen before they were able to do blood typing what would happen to the pregnancies where the mother and fetus had different, RH factors?
There would be complications. Some of it would be, sometimes it's rather mild, sometimes it would be severe. Like you would have the fetus would be, so it's called a hemolytic disease of newborn. So the fetal blood would be like have cell damage to it. I'm not sure if it's fatal all the time, but I know it's fatal some of the times, and like they wouldn't know why. So, but it doesn't occur that often, as often as you think it does, because honestly, most of the people in the world are RH positive. So the chance of having an RH positive, but like negative mother and an RH positive father is not as common as you think. And then for the second child, but yeah, it used to be a problem. In fact, that was one of the things. If you know about anything about Henry VIII, is that he had major issues conceiving children and shit like that. And I think that was actually one of the scenarios that they were thinking about, that this was what caused him to have so many issues.
Wow. So in the blood type, like if you're A negative, that's your RH factor is negative, right?
Right.
Wow. So does that apply to animals too, or just humans?
It definitely applies to animals, but animals have their own. Different animals have their own blood types and all this other stuff. Like I know apes have the ABO blood typing as well. I know rhesus monkeys have the RH factor as well. In fact, that's what RH actually comes from, rhesus monkeys. That's where we discovered it and stuff like that. So, but as far as how unique different animals are, I really don't know. Okay. Does that make sense to you guys? Does this make sense to you guys?
Yes, thank you.
Oh, okay. One of the things you guys have to kind of think about a little differently. Plus and minus are not the opposite of each other. Plus is the presence of something. B is a complete lack of something. You can't respond to nothing. Does that make sense? You can only respond to something. So if you are negative, then you can respond to a positive RH factor. If nothing can respond to a negative RH factor. So this is both yes. Okay. Does that make sense? Yes. It's like think of the negative as like type O. You can always get it. Okay.
Sorry, random question. Is
Yeah.
it possible that if a woman is pregnant with fraternal twins, that the babies would have different blood types?
Fraternal twins? Yes, absolutely.
So is that a danger to are the babies a danger to each other?
No, because they're not going to share blood with each other.
Okay.
Fraternal twins each have their own placenta.
Okay. Wow, that's crazy.
Yeah. Yeah, they have their own placenta, so they have their own independent blood supply. So they're not going to contaminate each other and stuff. Yeah. Well, it is very neat. Sometimes you absorb your twin And it happens more often than you think. All right, any other questions? Okay, so share my sharing. Let me turn this off first. Okay. Oh, oops. Damn it, I was on the right thing. Hold on. All right, so the objective, measurable. Okay. Okay, what is that?
I can't see anything.
Symptoms?
Oh, it's in the chat.
It's signs or symptoms. So I'm going with sign.
Okay, sign or symptom.
Symptoms.
Nope.
That's
It's a sign because you
signs are
can
measurable.
evaluate it
Yeah,
by observing it. Mhm.
it's objective. Okay, symptoms are subjective. Okay. Okay, I was talking about this a lot today, so you guys better know what it is.
Antagonistic something.
Bacterial antagonism.
Very good, Dr. Microbial antagonism. I'm sure you can figure that one out. Okay.
Opportunistic.
Yep. Very good. All right. Ooh, this is hard. Okay. All right. So you guys should choose one of the answers.
Is it coagulase?
Nope.
Because
Coagulase is the opposite. They cause blood clots. Okay.
Kinase.
Kinase, yes.
Kinase, sorry. You know I say things how they look.
It's okay. All right. But yeah, kinases, dissolve blood clots, coagulases coagulate, so causes blood clots. Okay. Bacteria that have one will have the other. Okay. So if you cause a blood clot, you have to find a way out of it. So like, does that make sense? Okay. Okay. So in other words, what does the first word mean?
Inside. Endo means inside. Genus is coming in from, I don't know.
Coming from inside you.
So it's like the person's own like natural stuff. They own like flora.
Yep. But good job bringing the word down. Okay. Wow.
Hi, little baby. She's behind you. She's too cute.
Charlie, what's up? Come here. Yeah. Then close it, okay? One more. Okay.
Did you say your daughter's name's Charlie?
Yeah.
Oh my gosh,
She's
that's
so cute.
That is real. Yeah, she's way cute.
What's she asking Santa for this Christmas?
Makeup. Nail polish to be precise. Yes.
Is Santa gonna bring that?
Huh? Maybe. I'm thinking about it. All right. What is this?
Will that be the prodromal stage? I was torn between prodromal and acute, but acute is like
It is
as soon as it happens, but during the incubation period, that can happen like days after you've been introduced, right?
yeah. So it goes incubation period, okay, and then prodromal stage, which you're kind of you're starting to feel like crap, but there's no like obvious signs or symptoms yet. And then the acute phase is where you're really kind of sick, and then the convalescent stage where you're recovering. Okay, does that make sense?
Got it. So the stage of infectious disease where specific signs and symptoms are seen and
Yeah.
the pathogen is at its peak. So that will be the
Acute.
acute phase.
Yeah.
Got it.
Yeah.
Okay.
The what gives it away? Specific signs and symptoms are only seen at the acute phase. Does that make sense? And then the prodromal is like you just feel kind of like crap, but not like feeling like crap isn't a sign or a symptom of anything. Does that make sense?
Got it.
Okay. All right, this should be really easy, actually. Like, which one of these is not like the others?
Ribosomes.
Yep. Okay, all the others can cause diseases, right? Or cause virulence or illness, right? Okay, so what's the difference between endotoxin and exotoxin?
Isn't endotoxins what goes in your body or whatever you consume, and then exotoxins is what you release from your body?
No, that is very, very much what it sounds like, but no, because endotoxins are actually incorrectly named, but which is part of the reason why for the confusion. But endotoxins, okay, are only found on the cell wall of gram-negative bacteria. Okay, the purpose of an endotoxin isn't really to do anything, but the gram-negative bacterial cell wall, when the bacteria dies, will release all this stuff, okay, lipopolysaccharides, and it will cause, in some people, it will cause a very severe inflammation, very severe inflammatory response.
So
Okay,
you only see the endotoxins like right at cell death.
yeah, you only see endotoxins at cell death. Okay, so if you have a gram-negative bacteria infecting you and you treat it with an antibiotic, and you can potentially cause a severe endotoxin response because you're killing all the bacteria and they're releasing all the endotoxin as they die. Okay, does that make sense? Exotoxins
Yeah, that
are
makes sense.
toxins that are purposely made by the bacteria in order to kill you or injure you in some way. Does that make sense? That's the difference. Okay.
So with the exotoxin, would they act like the same as like proteins do?
Yeah, they are proteins. They're proteins that are designed to harm the host. Does that make sense?
Got it. That makes a lot of sense.
All right. Okay. So let's see if we got it. Okay, so let's see if we have to answer this question first, and then I have more questions based on this.
Oh my,
Oh my, good. Okay, so what are the other questions, the other answers? So what is a carrier?
That's the person that's actually infected.
No. Carriers are infected, but that doesn't mean it's a carrier. So what's a carrier?
Isn't that like a person that passes on to another person?
Be more specific.
So like if I got the measles and I'm in your presence during my infected period
you're
and
not.
I pass it on to you, I wouldn't be the carrier of that?
You would not be a carrier. You would fail the class because you gave me measles, but you would not be the carrier.
I promise not to give you measles. I've already had them as a kid.
All right, but carriers are by definition not asymptomatic.
Okay,
That-
so is that like when the disease like live in you already, but you don't know you have it like?
Right, you don't have any signs or symptoms of it.
Got it. Okay, okay, okay. So the vector would be the person that's passing, that harbors it.
Vectors are ways of transmitting the disease, right? Okay, so next thing, what is a reservoir?
Isn't that where it lives, like where it's held, where it's growing, where it's living, where it's harbored?
Good. Does that make sense? So like COVID-19, the reservoir, COVID virus is the reservoir is bats, right? Because there's a lot of them that have it or something like that, usually inside infectious. Okay. Does that make sense? Yes. Okay. Okay. So what is a vector?
So this one I won't forget because you gave this one to me when I gave my oral presentation, and and I got it mixed up with, well, not really mixed up, but the virus that the African sleeping sickness, the Tessie fly was the vector. It wasn't the source. It was the person that carried it over to cause the problem, but it didn't originate in the Tessie fly. It originated from the trypanosoma bacteria.
Trypanosoma. Yes.
Yeah, that.
Does that make sense to you guys? The vector doesn't cause the disease, okay, but is aids in the transmission of the disease. Okay, does that make sense? So fomite is actually an animate vector. So okay. Next question. Nice. Okay, so some definitions.
Is it prevalence and incidence?
Well, it's an incident. Yeah. Okay. All right. So, okay. Okay. So what is mortality rate?
It's like referring to the deaths caused by the disease.
Good. What's the morbidity rate?
Morbidity is like when you suffer like from a disease or something.
Right. The number of people who are suffering from the disease, so sick from it, but not dead from it. Or, well, if you like, morbidity rate is always some percentage of the mortality rate. So does that make sense? So, like, obviously, if you die, you suffer from it, and stuff. So does that make sense? So mortality rate is going to be a small subset of the morbidity. Okay. Okay, these are all pretty easy. Which one? I think it's all one chapters. Which one? Bunch of chapters. Okay, good. Okay. So the antibody.
Is it the variable region?
Very good, the variable region. Okay, all right. Okay, that's this is it.
Plasma cells.
Plasma cells, good job. Okay. Okay, so what do cytotoxic T cell or, oh, okay, so sorry, let me try to paste this. Okay, so what do cytotoxic T cells do?
Don't they like kill the target cell?
What target? What do they kill?
Like the, wouldn't it be like like the actual thing, like actual antigen?
Okay, you can't really kill an antigen, you can only kill cells. So what cells does the cytotoxic T cell actually kill?
The infected cells.
The infected cells. Good. Does that make sense? Okay. The cells that are infected by virus or intercellular bacteria. Okay. Yes. All right. What about helper T cells? What do they do?
Don't they like help you like respond, like give you like a sense of immunity? Like it's like if you get something, it gives you like the know-how.
Like adaptive,
I'm sorry, what?
like an adaptive immune response.
Well, all of these cells are involved in the adaptive immune response. Okay. B cells and T cells are definitely involved in the adaptive immune response. Okay. So what makes a T helper cell?
They aid in killing the infected cell also, right?
I'm sorry?
They aid in killing the affected cell also, right? They aid in targeting the infected cell too, right?
But they don't actually kill the cell, right? So what do
They
they
activate
actually
T
do?
cells.
What?
They activate the cytotoxic T cells.
Good. They activate the cytotoxic T cells and they activate the B cells to turn into plasma cells. Okay. So they're in the center of the immune response. Okay. More than any other cell. They activate both the B cell response and the T cell response. Okay. Okay. Oh. Wow. Okay. That question didn't come out very well. Okay. So let me first put the question. Okay. Answer number one. Answer number two. Answer number three.
Answer 2.
Answer 2. Good. Okay. So immunological methods always determined by antibodies. Okay. Okay. So answer 1 is just genetic sequencing. Answer 3 is the phenotyping, the phenological explanation, something like that. Okay.
Hypersensitivity.
Good. Hypersensitivity. Okay. So what are the 4 types of hypersensitivity? So what is type 1? Yeah. All right. So let's review all these hypersensitivities. It's a hard thing. Okay, type 1 is the typical anaphylaxis response. So eating shrimp or breathing in pollen, whatever. But type 1 is a typical anaphylactic response. Okay. You have IG, IgE antibodies in your bloodstream. They bind to the antigen. They trigger mast cells. Mast cells trigger a lot of histamine release, etc., etc. Okay, that's type 1. Type 2 is cellular. Okay, you have antibodies to foreign cells in the body, and they will attack the foreign cells in the body. Okay, so that the example of that is like transplants or incorrect blood typing. Okay, type 3 is things that are dissolvable in plasma. Okay, small little things are dissolvable in plasma, but also mediated by antibodies. And what they do really is they cause localized responses like lupus and rheumatoid arthritis, where the antibodies respond to a soluble antigen. Okay, last but not least, type 4 is delayed hypersensitivity. Okay, that alone is caused by T cells. All the first 3 is all antibody related. Type 4 is caused by T cells. Okay, and therefore it takes more time. So delayed onset hypersensitivity, like poison oak, poison ivy, or like the TB response. Okay, the TB test response. Okay, does that make sense? Yes.
Can you say the name of the last one more time, Dr. Poon?
Delayed type hypersensitivity.
Okay, thank you.
Mhm. Okay. So yeah, okay. So that ended up being exactly all your questions that I just explained. So that involves B cell immunoglobulins, so antibodies, is type 1, 2, and 3, but not 4. 4 is T cell related, so delayed type hypersensitivity. All right. Okay. Let's see. Yeah, so this. All right.
Is it type 4?
4. How do you know type 4?
Because of the delay.
Right. Okay, because it's at 48 hours. It's a typical type 4 response. Okay, this is going to be a hard question.
Graft versus host.
Good. Graft versus host disease. Good. Okay. Does that make sense? Yes, that's usually caused by bone marrow transplants. Okay. Okay. Easy question.
Hypersensitivity and autoimmune.
First one's right. AIDS is not an autoimmune disease, right?
Immunosuppressant.
Yeah. Immunodeficient. I'm pretty sure you figured that out from the examples. All right. Does that make sense? Yes. Yes. Okay. Cool. Okay. I think. No. No. All right. Any other questions? So I went over a bunch of like most of the. I skipped all the easy ones. So like the rest of them are relatively easy. All right. What? Okay. Cool. All right. You guys are flirting with each other, so we're done. Now, okay. All right. Any other questions before I let you guys go? All right. Yeah. Just, I mean, just do the practice exam bunch of times and stuff like that. It should be this. You can do the practice exam more than once, right? So if you do a bunch of times, you should be able to see all the questions. Okay. All right. Any other questions before I let you guys go? All right. Okay, bye,
Yes, Dr.
everybody.
Poon, I have a question. Sorry. For the whoever's presenting, are we presenting on Thursday after the exam, or are we presenting
No.
next week?
There's, I won't have you guys present next week, because there's no, like, after you guys finish with the exam, first of all, I don't want you guys worry about presentation before the exam. And secondly, I want to, like, by the time you finish the exam, there won't be much time to talk, so this will take all of it. So, honestly, you guys will have to end up presenting, recording yourselves and stuff.
So
That's
real
good.
quick,
Thank you
Dr.
so
Bloom,
much.
for some clarification, was Tavon right? Somebody asked it in the chat earlier, but I think I messed this all up. So for the peer reviews,
Yes,
we have to do a peer review per week.
yes,
So it's a total of 3 for us then.
yes.
Okay, my boy T was right. All right. Yeah, I'm all messed up on them damn peer reviews. My anatomy peer review winded up on my microbiome post. I mixed that all the way up. I don't even know how the hell that happened, but all right.
All right, cool. Okay, bye, everybody.
Thank you,
Bye.
Professor.
Yeah.
Bye, thank you.